Okay, we've spent a lot of time looking at functional groups in organic and biological chemistry that have oxygen in them. Now we're going to look at some of the nitrogen-containing functional groups, and they are amines, amides, and basically amides go together to make proteins. Let's do it. So, as we often do, we'll start with a little bit of stuff about naming. I've picked an interesting one here. This chemical is commonly known as putrescine. Putrescine is one of the odours given off by dead and decomposing bodies, but it's also, as I've talked about in class, found in semen, and one of the things that gives semen its characteristic smell. So let's have a go at naming this compound. We've drawn it in the skeletal type structural way, so we can imagine that there's a carbon there, there, and there. So we've got 4 carbons. So it is a bute for 4. It is an amine. So it's an amine, but in this case here we have 2 amine groups. So we would say it is a diamine, and they are on the 1st carbon and the 4th carbon. So that would be butane 1,4-diamine. Okay, now we've already talked about primary, secondary and tertiary alcohols, and now we're going to talk about primary, secondary and tertiary amines. And it's similar but a little bit different. In a primary amine, the nitrogen is only bonded to 1 other carbon. Therefore, there's 2 hydrogens. In a secondary amine, the nitrogen is bonded to 2 other carbons. And in the tertiary amine, surprise, surprise, the nitrogen is bonded to 3 other carbons. Now, nitrogen, if we go right back to the basics, has 5 electrons in its outer shell. It wants to get to 8, so it wants to share 3. So it tends to bond to 3 other atoms. But we get left with this lone pair of electrons that's also on the nitrogen. Now, that lone pair are often quite happy to accept a hydrogen ion. So, because they can accept a hydrogen ion, that makes them a base. That's our definition of a base. And when they accept that hydrogen ion, we get left with this having an ion. We can sometimes put the charge outside the brackets. I tend to just put the charge there next to the nitrogen. So we end up with this positive charge on the nitrogen once it accepts a hydrogen ion. So again, it turns it from a neutral compound into an ion. Why is that important? Well, again, it's about solubility. The amine form here can hydrogen bond with water, so it's reasonably soluble, but the protonated amine form can form ion dipole interactions with water, which are even stronger. Therefore, that makes it more soluble. There are a number of drugs that contain amine groups that often are given to us as treatment in the protonated form. Because they'll be more soluble, our body will absorb them much quicker. They'll get into our bloodstream and do their job as a drug much quicker. So a common application or context of this sort of question on the solubility of amines relates to the solubility and absorption of drugs into our system. Okay, that I think is all we need to know about amines. Let's keep moving. Okay, let's talk about amides. Don't actually think we need to name amides, but just worth pointing out, amides are a lot like esters, but instead of having an oxygen here for your joining group, you've got an NH here. So in many ways we can think of these as the nitrogen equivalent of esters, and a lot of the reactions they undergo are going to be very similar to the reactions we saw of esters. So, for example, to make an amide, we add together a carboxylic acid, but instead of an alcohol, we add it to an amine. So, again, it's a condensation reaction. So, if we, just get the right pen, we basically lose and just one of those hydrogens, because we still get one hydrogen left attached to the nitrogen, and we get a water produced here. So again, it's a condensation reaction. Carboxylic acid plus an amine equals an amide, as opposed to a carboxylic acid plus an alcohol equals an ester. Again, if we take a diamine and a dicarboxylic acid, we can make a polyamide. We did that just before, or previous, I'm not sure, it might be in the previous video actually, with a dialcohol or a diol and a dicarboxylic acid. An example of a polymer made like that is nylon. So, nylon is made from a dicarboxylic acid and a diamine. Again, we can break them apart, and again, we get different products whether we break them apart under acidic conditions or alkaline or basic conditions. So, we break an amide under acidic conditions. Again, it's a hydrolysis reaction because we're adding water. We form the carboxylic acid. It's under acidic conditions, so the acid will be protonated. And we'll form the protonated form of the amine because that lone pair of electrons on the nitrogen will accept a hydrogen, giving it that positive charge. If we do it under alkaline or basic conditions, we form the carboxylate ion, where the carboxylic acid has lost its hydrogen, it's donated its hydrogen to a water, and in that case we just form the amine, which is, if you like, the base form of an amide. Think of the acidic form as the protonated amine. So yeah, that is amides in 2 minutes and about 40 seconds. Cheers. Okay, into the final part of proteins. And before we talk about proteins, we need to know what an amino acid is. An amino acid could really, if we wanted to, be called an amino carboxylic acid, but we say amino acid for short. But basically what it does is it contains an amine group and it contains a carboxylic acid group. It's just drawn in the condensed form here. I've got one over here, an amine group and a carboxylic acid group. Now we know that when we form a amide, which we also often call a peptide link, just a different word, an amide link, a peptide link are the same thing. Biologists tend to more use peptide link. They are formed when an amine reacts with a carboxylic acid. So, these amino acids, if you like, are like little Lego building blocks. They can be joined together really, really easily. And this is what happens in the ribosomes in your cells. For those of you who do biology, that process of translation, where the RNA is read and all the amino acids get put together in the right order to make all the proteins that make up so many of the molecules that make up your body and keep your body functioning. So we've got a list here of just a selection of 6 of the more common amino acids in our body. There's basically about 20 amino acids. You'll often hear of bodybuilders and the sorts taking amino acid supplements to give themselves, I guess, more building blocks for the muscles that they need or are trying to build up. Although I'm not sure how much science is behind some of that stuff. So, before we get into the forming of proteins from these amino acids, just a few words about amino acids. We have basically an acidic group here. So if we would put this into basic conditions, we could lose this hydrogen, because this is an acid, if you like. This part acts like an acid. It could donate a hydrogen. However, this part, the nitrogen has those lone pair of electrons. It acts like a base and it can accept a hydrogen. And what amino acids will often do is what we call self-ionize. So the hydrogen from here will get donated to the nitrogen here, leaving a negative charge on this end of the molecule, a positive charge on this end, and we call this a zwitterion. So This is what we're talking about here, with amino acids have both acidic and basic properties. The acid can donate, the base part with the nitrogen can accept, and they can self-ionize to form these Zwitter ions. So you need to be able to draw that structure for any given amino acid. Now, amino acids can also, well, they just form in a very similar to the reaction we've seen. When we get a carboxylic acid and an amine, that's when we form an amide. So all of these can link together with just one of those hydrogens. One of those hydrogens, in each case water is released, and so on. And they all join together to form our, whether we call it a polypeptide, or another word for a polypeptide is a protein, or it's basically a polyamide chain. Really all different words to say the same thing. So that's how we get this structure form here. Again, we need to be able to identify, well, sort of identify repeating units. But the thing is, in amino acids, we don't have this really regular structure of, like we would in a polymer. We can have lots of variation in this chain of amino acids. And what's different, it's just what's on the bottom. What these R groups are here. And just looking at that, I better fix that up. That should be an R double prime. But basically, here, what's on the bottom of all these amino acids, often we call the tail. So we refer to the amino acid where the amine and the carboxylic acid is as the head, and this part down here is the tail. So we get this continuous chain. And we're going to talk about this more in the next part. So for the biologists, this will start to sound like something you've heard before. The proteins, the amino acids go together to make the proteins. We talk about 4 levels of structure in a protein. Firstly, we talk about the primary structure. That is the sequence of the amino acids. So that is the order of those amino acids that make up the protein. Secondly, we have the secondary structure, which is the localized structure of the protein due to hydrogen bonding between the peptide links in the protein, and tend to either get alpha helix or beta pleated sheets. So those heads that I just talked about of the amino acids can all interact together to form various localized structure, whether it's a helix or the beta pleated sheets, and that gives you the secondary structure. Finally, the tertiary structure relates to the way the protein or the polypeptide chain folds, and this is caused by the interactions between the tails of the amino acid groups. And then finally we have the quaternary structure, which refers to how 2 or more polypeptide chains weave into each other, or how they link up within a protein that contains more than one polypeptide chain. Now, the arrangement, the spatial arrangement of a protein is what gives the protein its biological activity. Structure is normally the important thing. For a protein, it's the shape of how it binds to a receptor, or how it pulls apart 2 molecules as it acts as a catalyst, all that sort of stuff. So, if we change the pH or temperature, we can actually cause some of those secondary bonds, particularly between the tails in the tertiary structure, to break. When those bonds break, that causes the shape to break. When the structure breaks, it loses its biological activity because the shape has changed, and it is the shape that is crucial to the functioning of a protein. So, I think at least everyone I'm teaching at the moment does biology, so that stuff should all sit very neatly with what you've learnt in biology. Cheers.